Hello, welcome to 13 Universal Learning Beyond Barriers, and this is A-level CIE Biology with Shedro. Now, guys, we are starting a new series called All Topics Theory, Paper 2. This is Paper 2 Biology, okay? Series 1. We have been doing MCQ. Now, we want to start doing Paper 2 Theory series, okay? So when we are done with this paper 2, we go to paper 4, paper 5, 3. I just want you to stick with us. All right, please, I want to say this, that all the resources are from CIA past papers, okay? The past questions and the math scheme. All right, get your writing pad. Remember that what we do here is that we practice, we solve questions, okay? So if you're on this platform, you're watching this video, that means you must have gone through the question, the topic. You now want to do application. Are you ready for application? Are you ready? Come with me. Now look at the first question. Now that is the first question there says, look at it there. It says the malaria, that's not material, it's a typographical error. The malaria pathogen, Plasmodium falciparum, enters red blood cells after a person becomes infected. After some time, each cell of P. falciparum divides to form daughter cells. You can see that. Okay, see, it's forming daughter cells here. All right. Okay, let's go on. The figure 1.1 shows a cell P. falciparum that is forming many daughter cells. Okay, now let's go to the question. The question says, with reference to fig 1.1, suggest how the presence of P. falciparum affects a red blood cell. Okay, how does the presence of P. falciparum? Now, can you see, guys? This is the P. falciparum inside a red blood cell. This is red blood cell. So one of the things you do in solving questions is that you have to pay attention to stimulus material. One of them is the diagrams, okay? And that one is the text, the diagrams. So you see they've shown us cell membrane of red blood cells. See the cell membrane? All right, so inside of it is this Plasmodium falciparum pathogen. Okay, see it multiplying, dividing, having a nice time, okay? Dividing. So the question says, with reference to fig 1.1, suggest how the presence of P. falciparum affects red blood cell. Now, if you look at this, guys, how does it affect it? Suggest. You can see that is changing the shape of red blood cell. All right, is changing the shape of, you know, red blood cell has a biconcave disc shape, which gives it large surface area to take up oxygen. All right, so it's changing the shape. So the shape changes by concave disc shape of red blood cell. That's the first point you're going to mention. Now, this is a 3 marks question. Sorry, it's not there, but it's a 3 marks question. Is that okay? All right, so the first point, looking at this, all right, is that it changes the shape. This doesn't look biconcave disc shape, right? It changes it biconcave disc shape of red blood cell. Number two, secondly, you can see that the Plasmodium parasite, okay, divides and then causes red blood cells to expand and then burst. So it causes red blood cell to burst. That's another effect, how it affects red blood cell. So red blood cells will burst, okay, and when they burst, the other effect that follows it is that it will release hemoglobin, okay? So hemoglobin is released when it bursts, okay? Now, don't forget that this parasite also feeds on hemoglobin, all right, and then reduces the amount of hemoglobin. But hemoglobin also can be released when this bursts, and when this happens, okay, so less oxygen will be transported. Okay, so I've mentioned it. All right, so let's look at the answers. Let's look at what the math scheme says. Okay, please remember that everything that's said here, you can derive your answer by looking at the text and what, and the diagram here, because they say with reference. Okay, let's see. So it says irregular shape of cell. So you can see that the shell is having irregular shape. Okay, or if you don't want to say this, a cell loses its biconcave shape, so it becomes irregular. That's the first effect. Okay, the second effect, okay, the another way to say it is that cell swells, so it's the same point, or cells in larger, or cell expands. Okay, but we know that it changes the shape. All right, Now I said ignore cell becomes flaccid. Now because flaccidity or Turgot deals with osmosis, there's no osmosis going on here, so there's no osmosis. Okay, so this is the first thing we'll see here, and any of this, any 2 points, all right, from the following. So you can see that cell membrane burst, you're going to burst the cell membrane, or cell burst, or cell dies. Okay, and that's cell lysis, hemolysis. Do you get? And then we have because of that, cell is going to have shorter lifespan. That's the same thing. Okay, if it bursts, that means it's going to have shorter lifespan, and then daughter cells of this hemoglobin will fill the cell. Okay, the same thing. Cell loses hemoglobin, or hemoglobin used by pathogen. Pathogen uses amino acid hemoglobin because it needs to make its own protein, so it's going to use the amino acids from hemoglobin, digest hemoglobin, uses amino acid to make its own protein. All right, so therefore less oxygen will be transported by red blood cell. Okay, less uptake of oxygen. All right, so of course this is a pathogen going to release toxin. Okay, so that is it, guys. All right, so it says ignore changes in surface area to volume ratio. So it's still the same surface area and the same volume. All right, so these are the any of these two, okay, answers this question. Okay, let's see what you think. Let's go to the next question, guys. Okay, yeah, so let's go to the next question. Yeah, the next question, the same question here. We're going to question 2 of it. All right, look at the question. That's B. This is 1A, 1B. So 1B says when P falciparum divides, there is unequal division of the cell. Yes, can you see? Unequal division of the cell, and you see, yeah, see, unequal division of the cell. This is larger than this. All right, of the cytoplasm to form small genetically identical daughter cells. So genetically identical daughter cells. Now the cytoplasm are unequal, but they are of the same, all right, the same, they contain the same genetic material. Okay, so the question says outline the events that occur in the cell of P. falciparum to form the daughter cells. Now guys, shown in fig 1.1. Don't forget that P. falciparum is a eukaryote. All right, it's a eukaryote, so it undergoes the, because we're talking about how it divides. Okay, so because it is eukaryote, so it's going to undergo the mitotic cell cycles. You know, do you remember the cell cycle? Yeah, see the cell cycle here. Don't forget that these are the events it will undergo. So this is mitosis. Of course, that is cytokinesis. That is G1. Okay, that's S phase, and that's G2. So this is G2. All right. Yeah, this is mitosis M. Okay, this is cytokinesis. This is G1, and this is S phase. Okay, yeah, that's S phase. So you can see that, right? So you see, so they're asking us outline the events. Okay, and if you check there, you see that this is about 3 marks question. Okay, so outline the event that happens. So guys, so this cell will have to grow bigger. So it starts from G1. So these are the events. The cell grows bigger because this is start from G1. It goes like this. All right. The cell grows bigger. All right. So it makes more proteins. All right. So there's increase in size. More organelles are being made. Okay. Now, of course, you go to the next stage. All right. We have semi-conservative replication of DNA occurring in S phase. Okay. So more synthesis of what is it called, proteins, okay, occur here. More ATP synthesized. All right, organelles made. Then you go to mitosis. These are events. So in mitosis, it goes through prophase, anaphase, metaphase, anaphase, and telophase. So you have to mention, all right, it goes through this event. Prophase, the chromosomes coil up, and then in anaphase they line up along the equator. Telophase, anaphase, the sister chromatids separate, and telophase, of course, 2 nuclei are formed. Okay, these are the events it goes through. So let's look at the answer. Any 3. So it's a pathogen feeds and grows, synthesizes. Okay, so this synthesizes means it's going to make chemicals, proteins. So it increases in volume, of course, mass of the cytoplasm, because it's going to grow big. It's going to grow big before it can divide. It doesn't grow big, it cannot divide. So production of organelles, okay, is a eukaryote, maybe mitochondrion, okay, makes organelles, and then DNA replication occurs. Once it's done with this stage, enters this stage, DNA, semi-cosmic replication of DNA, okay, reject incorrect phase, so you have to follow it. Mitosis, 4 named stages, okay, production of several many nuclei, cytokinesis, yes, we have to mention cytokinesis is going to occur, and then of course, yeah, so ignore division of food, then total cells board off, see the total cells boarding off here, okay. So this is the, because they say outline is sequence, okay, just states what happens in a sequence. Is that okay? So that's the end of this, of course. That is it. So let's go to the next question, guys. All right, any question here? All right, look at the next question. It says, in 2023, the World Health Organization set a target for researchers to create a vaccine for malaria. WHO require vaccine to show 75% efficacy and be ready for use by 2030. We're in 2024. All right, so they're working on this project. Efficacy is a measure of the effectiveness of a vaccine in reducing the number of new cases of malaria. A trial of R21 matrix MTM, okay, vaccine in Burkina Faso in 2020 achieved 77% efficacy over a 12-month period. Wow, that's commendable. Now it's proceeded the target. A control group received a vaccine for rabies. Okay, vaccines stimulate an immune response with the production of antibodies. Okay, let's see what next. Okay, the question says, that's 1C. All right, that 1C says, explain how antibodies will reduce the spread. Okay, explain how antibodies reduce the spread of the malaria pathogens through bloodstream. Guys, now if you have an idea of how antibodies function, it's going to help here. You notice that antibodies are specific to antigen. So antibodies, because they say explain means give us reason, okay, on how it's going to happen. So antibodies play different roles, okay, in providing immunity. The first role they play is that antibodies bind to specific antigen on the pathogen. All right, and when they bind to the antigen on the pathogen, they stop the pathogen from spreading. Okay, they clump up the pathogens and then stop them from spreading. Another way they also provide immunity is that they could also help to facilitate or stimulate phagocytosis. Now, such antibodies are called opsonins, right? So opsonins, they mark out pathogens, or they label them for phagocytes. So how do they do that? Because we're talking about how do they do that? So they bind to the receptor on phagocytes using their constant region, and then use their variable region to bind to antigens on the pathogen. Is that okay? And then allows the phagocyte to engulf them together with the pathogen. Okay, and you know, some of them they act as antitoxins, so to neutralize the toxin made by the pathogen. Okay, and of course, let's look at what the mask NA3. So antibodies bind to antigen. That's how it works. It binds to antigens. All right, epitopes. Why? All right, because they attach, combine. Okay, why does it bind to that? And of course, when it binds to that, it forms antibody antigen complex. Okay, this is the sequence. So this is the masking, Cambridge masking. Accept forms antibody antigen complex. All right, and then ignore antibodies bind to pathogen. It doesn't bind to the pathogen, it binds to the antigen on pathogen. So the antigen is on pathogen. All right, and again, antibodies, all right, you know, specific antibody is binding to a specific pathogen. All right, complementary shape, yes, it's complementary to it on surface of malaria pathogen, so that's why it's binding to it. So it prevents pathogen entering red blood cells, liver cells. Okay, help facilitate destruction by making pathogens for phagocytes. Okay, look at that. Stimulate phagocytosis. Now, antibodies that do that, they're called opsonins. The process is called opsonization. They coat pathogens, all right? So phagocytes have receptors, all right? That's how for constant region of antibodies, of course, and of course agglutination and all of that. Please don't mention T lymphocyte, T cells, T cells do not make antibodies. All right, this is humoral immune response. It's not cell mediated. All right, guys, let's go to the next question. Some vaccination programs have been more successful than others. Discuss the factors that contribute to the success of a vaccination program. So if a vaccination program is successful, okay, so what are the factors, guys? I want you to pause the video and try to solve this question, like our drills are, our normal drills. Pause the video and solve the question. Of course, if it's going to be successful, what are the factors? Say we should discuss it, right? Number one, I feel, okay, I think is one of the factors is that if for vaccination to be successful, that means that the vaccine is thermostable, okay, it's not destroyed because of temperature, all right, or the pathogen does not mutate. So if pathogen mutates, it's going to create a new antigen, and that will require another vaccine, right, because to be made in order to provide immunity. Okay, now another thing is that for it to be successful, it's possible that more people were reached, more people were available for vaccine. Okay, and then more people, they were reached because they were also educated on the purpose of the need for vaccination. Do you see that? So we had more people trained to deliver the vaccine. Okay, and again we have not everybody would have been vaccinated, right? So there's what is called herd immunity was achieved. The herd immunity in this case talks about more people, large proportion of population vaccinated, okay? And when large population of people are vaccinated, it reduces, all right, the transmission of the pathogen, especially, all right, it reduces it, so that it doesn't affect the vulnerable people in the society. All right, so let's look at what the answer says. As answer, any 5, access to whole population, yes, all people at risk. So many people, the whole population were vaccinated. That could be a reason. Number 2, either they're reaching enough of the population to give a herd immunity, right? Herd immunity must be qualified. So ignore. So large proportion of people were vaccinated so that others that were not vaccinated were protected. Okay, because it reduces transmission. Now, vaccination of children, yes, children were vaccinated early in their lives, okay, before time when they're more at risk. For, of course, people educated, right, importance, benefit, advantage of vaccination. Now that they were able to counter anti-vaccination campaigns or misinformation, so that people are willing to take vaccine, right? So long duration of protection given by, so the vaccine provided longer duration. Okay, all right, reference use of live vaccine, yes, could do that. Little or no mutation of pathogens, little or no antigenic shift, few or no strains developed. You can see that. All right, ignore resistance to vaccine. There's no resistance to vaccine or such a thing. Ability to change vaccine in response to changing strains. So ability to, all right, in case mutation occurred. So there are plentiful or supply, enough supply of vaccine. There are a lot of information here and I'm asking. Sufficient number of people trained to deliver vaccine. You can see that. That is idea that administering vaccine is simple process. Any positive reference, cost production, transport, storage, all right. So these were cost effective, all right. And of course, vaccine is provided free. Long shelf life of vaccine, stable vaccine in high temperature. Vaccine can be freeze dried. Okay, no booster required. You see, easy to find people who need boosters. Government health authorities must have enough money to fund it. All right, use contact tracing to find people at risk of the disease. So these are the provide incentives. Okay, there are a lot of reasons. Okay, let's go to next question. No side effect. Okay, this is question 2. Now this question 2 I think is in biological molecule. It says glucose is used in the synthesis of amylose. Glucose is first converted to glucose 1-phosphate G1P. Starch phosphorylase is an intracellular enzyme that can catalyze the synthesis of amylose from G1P, which is the substrate for the reaction. So this is glucose 1-phosphate. Okay, this is arrow here. It gives us amylose and phosphate. So this is a condensation reaction. Glucose 1-phosphate. This is a monomer or substrate. Starch phosphorylate is the enzyme. So these are the products. So you have to first of all understand the question. The rule of engagement states that do not solve any question you don't understand. Read the question properly understand the context, the information the questions provide. Okay, this is the result. You can see the result of the investigation as shown in fig. All right, in the figure, students used a calorimeter to investigate the progress of the reaction. Okay, so calorimeter measures absorbance. All right, now look at the next one. The student made a reaction mixture containing 0.1 mole per dm cube in a buffer solution at pH 6.0. You can see that a very small quantity of amylose was added to initiate the reaction. Solution of starch phosphorylase was added to the reaction, and samples were taken at 1 minute interval. Each sample was added to a dilute iodine solution, stirred, and then poured into a cuvette. The absorbance of each solution was recorded. You can see that, guys. This is time. So every 1 minute, they take a sample, pour it into a cuvette, okay, and they'll measure it. You can see that this absorbance. So with time, absorbance increases because more amylose or more starch is being formed. Is that okay? Can you see it's increasing? Why? We have more amylose. So because we have more amylose is being formed, so more light wavelength is being absorbed. Okay, so we have higher concentration of amylose. So the more concentration we have, the more we absorb, the more light wavelength is absorbed. That is why, of course, it reaches at 12 minutes. It has reached the maximum, which is 1.8. So 1.8 is 12 minutes, but it looks as if it's going to start leveling off. All right, at 1.8 absorbance. Okay, but let's see the question. Now they say explain why the absorbance increases as shown in fig 2.1. So why does absorbance increase? All right, can anybody help us tell us why does it what does absorbance increase? Of course, it increases because more amylose is being formed, more glucose is being formed. Is that okay? Yeah, so no, not more glucose, more amylose, more starch is being formed. So, and of course, more wavelength is being absorbed, so more concentration reaction occurring. All right, so look at it, answer here. More starch is produced, of course, so iodine becomes darker. Remember that this was put in iodine solution, so iodine changes from yellow brown to blue black. So blue black because more amylose or more starch is being formed, so more light is being absorbed. Is that okay? All right, let's go to the next question. So just 1 mark. The student took their final sample at 12 minutes. Predict the result for absorbance if the student had continued to take samples for further 10 minutes. Explain your answer. We have seen that, of course, the curve will level off, okay, to reach plateaus, okay, and that's the prediction, okay. It will level off. Let's explain your answer. Why? Because the substrate would have been used up, okay. Substrate would have been used up. The substrate here is glucose 1,6-phosphate has been used up, okay. All right, or you can say that the substrate becomes the limiting factor here. It becomes the limiting factor. All right, let's look at what the mass scheme says because it's a prediction. All right, absorbance remains constant, which is plateaus at 1.8. Okay, so that 1.8 was the maximum. So increases or flattens. Okay, it might increase more and then flatten. That's the explanation. All substrate used of commodity amylose. Calorimeter reaches its limit of absorbance. So guys, of course, this. So see, substrate concentration is limited. Or another way, prediction, absorbance increases, okay, less steeply. It's possible that increases less steeply or increase slows, all right, slows down. Yeah, that's going to happen. Explanation, you can see substrate concentration is becoming limiting factor. So that's going to happen, okay. So you can go back and see the graph. You see that when it continues to increase, all right, so the graph appears something like this, okay. So this is time here, just this absorbance, okay. So it continues increasing onto continues increasing, okay. But it's going to get to a point where it starts leveling off, right? So it's supposed to continue to increase slowly until it levels off. Because once the substrate reduces, it's going to level off, right? Okay, let's look at the next question. Okay, 2AI, state an advantage of using a calorimeter in determining the progress of the reaction. Yeah, if you use a calorimeter, you'll be able to get quantitative value, okay. Colorimeter measures the quantitative value, and then if you get the quantitative value of absorbance, you can use it to plot graph. Okay, you can also use it to determine concentration of a solution, concentration, all right, of a solution. Just get the absorbance, trace it on the curve, and then it gives you the what I'm trying to say, guys. So if this is a curve, all right, if you plot the curve, all right, and then against concentration, after plotting it against time, get the concentration, right, and absorbance. So I don't know the concentration of a particular um, solution, but I don't know concentration. This is concentration. So, but once I know the absorbance, I can trace the absorbance to the curve. So colorimeter helps me to do that, to plot a curve and be able to determine the concentration of solutions. Okay, and again, I don't have to use the eyes to observe color change because it gives a subjective result. So anyone from the following: colorimeter gives quantitative numerical results. Okay, idea that it can use values from the colorimeter to plot graph. Yeah, we've said that results are not subjective, so no judgment made by the eyes because of eye defects. Okay, so idea that can use calibration curve to determine concentration of starch. Yes, you can use that. That's what we just showed here now. All right. You can see can take continuous readings, do not have to take samples. Yes, so continuous reading. So don't use accurate, actual, reliable. No. All right, it's not the reason. So these are the reasons, guys. Okay, see the last question. I guess this the reaction catalyzed by starch phosphorylase occurs at the end of amylose molecules. Describe the sequence of events that occurs when a starch phosphorylase catalyzes the addition of a molecule of glucose to the end of amylose glucose. Remember that starch phosphorylase is an enzyme. They're just asking us enzyme, all right, mode of reaction. Okay, so right, remember the substrate here is glucose. So glucose binds to the active site. So that's what describe. When they say describe, sequence. So the substrate, which is glucose complementary, binds to the active site of this starch phosphorylase, and then ES complex is formed. Okay, ES complex is broken down, all right, to form product. Of course, you know that amylose has only unbranching. Okay, sorry, unbranching. Yeah, so only it will form 1,4-glycosidic bond. Okay, 1,4 will be formed. Okay, but if it is an induced, you can use induced mode of action. Okay, glucose binds to starch phosphorylase. Starch changes its shape to give us, all right, ES complex. All right, and then look at it. Substrate glucose, okay, binds to active site, forms ES complex. These are sequence of events. End of amylose binds to the active site, all right, together, and then we link two of them. ES substrate complex. Active site changes shape when substrate bind due to induced fit. Enzyme decreases the activation. Yeah, that's one of the action. Of course, role of enzyme is to lower the activation energy. Right, forms glycosidic bond, 1,4-glycosidic bond, and that reaction is conditional reaction. Water is formed. All right. Any detail of binding to the active site, formation of hydrogen bonds. All right. So substrate molecules put under strain. So what happens? All right. The temporal bond between R group of amino acid lining the active site and substrate, okay, could, all right, join the reactive groups to form, all right, the glycosidic bond. All right. Okay. So guys, thank you so much. I hope you enjoyed this first class on paper 2. All right, we're going to do series 2 in a short time. So share the video, make comments, ask questions, okay, and like our videos. Thank you. See you next class. Bye.